All right, so today and next week we'll be talking about cellular physiology and learning about the function of cells, along with looking inside of cells to find out everything about their internal structures and how they function. First thing I want to do is just define what a cell is. Basic unit of biology, therefore life. Biology is the study of life. Physiology is a subset of biology, so you can think of a cell as a basic unit of the human body function. Cells are living structural and functional units enclosed by a membrane. One of the principal theories in biology, and this applies to us as living organisms, is this modern cell theory, which has several basic principles. 5 things are listed here. All organisms, which we are an organism, are composed of cells and cell products. The cell is the simplest structural and functional unit of life, meaning that those various life processes that we talked about, like metabolism, responsiveness, reproduction, cells can do that. Our structure and functions are due to the activities of our cells. That's really the one I like to emphasize the most. When we talk in this class about the various functions of the stomach and the intestines, your heart, those functions are a direct result of the cells that make up those organs. Where does saliva come from? Cells in our salivary glands that produce the saliva. Various other things that I could get into and use as examples, but I won't. Cells come from only pre-existing cells, so they don't just appear out of nowhere. They come from a previous cell. We all began our life as a single-cell zygote. That's then become where we are today, trillions upon trillions of cells. And you could even compare our cells to, basic cells like bacteria or a fungus or any other animal. So, not that is really that important for this class. One thing that is very similar among all the organisms and their cells, except they're small, microscopic in size. You may have wondered why, maybe not. I'm going to talk about why, even if you have never wondered why. Three things limit the size of a cell. The requirement for an adequate surface area to volume ratio, which is the most important, because it affects the other two. It affects the rate at which molecules diffuse. Diffuse means stuff coming in and out of the cell. And then that's going to affect the need to maintain adequate local concentrations of specific substances involved in cellular processes. Basically, what we mean by that is our cell has to have specific makeup of various things in order for it to function correctly. And that is directly correlated to the ability to bring stuff in and out of the cell, which, if the cell is too big, its surface area to volume ratio is thrown off. Let's illustrate this with a figure here on the next page. We've got 2 different cells, and it's important to understand that cells are 3-dimensional, which is why I like this image. It shows that we've got them 3-dimensionally with their 6 different sides. Just like this classroom, which has 4 walls, a ceiling, and the floor, making it not a perfect cube, but a pretty close to perfect cube. Okay. You guys could all probably agree with me, or probably would all agree with me, that this cell here, that's 10 by 10 by 10, that's a perfect cube, is half as big as this one, which is 20 by 20 by 20. Okay. Well, when you look at surface area refers to basically the outer faces. How much of these faces there is. Okay. We've got surface area for our small cell, which you calculate as 10 times 10. So it's length times width. And since there's 6 faces, you times it by 6. The surface area of this small cell is 600 square microns. When we double the size of the cell, we go from 600 to 2400. So it's gone up 4 times. So the surface area has grown 4 times, even though our cell only doubled in size. Okay? That's not the issue. It's the volume that's the issue. Here, for our small cell, we calculate volume as length times width times height. That's 1,000 microns. That's cubed microns. Look what happens to it here. How many more times is 8,000 than 1,000? 8. 8 more times. And so we grew in surface area 4 times, but our volume grew 8 times. Basically what happens is the internal part of the cell gets way too big, and therefore the cell can't make sure everything is correctly concentrated because it can't bring enough stuff in or out to match that growth in size. Think of it if this room tripled in size. Could I reach the back of the room without like a microphone to talk? And would I be able to properly teach the people in the back of the room? No. And so having this smaller classroom, I'm able to capitalize on having a good surface area volume ratio. Okay. What we're going to do now is talk about the structure and function of the various parts of the cell. We'll pick up with this on Monday because we're not going to get through it all. We'll start with what you find inside the cell, which is its cytoplasm and cytoskeleton. Cytoplasm is a thick gel-like substance which includes organelles, like mitochondria that we've talked about today, nuclei and endoplasmic reticulum and ribosomes that we'll talk about on Monday. And they're suspended in a semi-fluid substance called cytosol. Cytosol is the fancier way, probably, to describe intracellular fluid, which we've talked about intracellular fluid since our first lecture, fluid within our cells. A lot of times you'll actually hear me and see me use cytoplasm and cytosol interchangeably. Technically, cytoplasm is the fluid plus the organelles, where the fluid is just cytosol, but for kind of this class, it's not that big of a deal to distinguish them. Now, the fluid of our cells, that cytosol, is far from being structureless. It is permeated by an intricate three-dimensional array of interconnected framework we collectively call the cytoskeleton. Just like our skeleton, it provides shape and support to our cells. Okay, it helps organize various things. There are 3 things that make up the cytoskeleton: microtubules, intermediate fibers, microfilaments. Microtubules are the largest. They are kind of a tube-like structure. Think like a PVC pipe or some other type of tubing in your house or apartment. It's kind of what they look like. They're actually made up of a protein called tubulin, so it's kind of nice. You got tube all over the place. Their structure, their name, the type of protein that makes them up. They form an organelle called centrosomes, which we'll see what those are here probably Monday. They help form the shape of the cell. They help organize the movement of organelles. They move chromosomes during cell division. If you've ever taken biology and learned about mitosis and meiosis, you might remember the things that look like spider legs that come and grab the chromosomes. Guess what those spider legs are? Microtubules. And then they'll form specialized cell projections, which we'll talk about what goes on. Intermediate fibers or filaments, thicker than microfilaments, thinner than microtubules, so that's why they call them intermediate. they're not made up of one standard protein. Different cells are made up of different proteins that make up these intermediate fibers. They help resist mechanical stress to the cell, stabilize the position of organelles, help the cells attach to one another. And then the smallest one are microfilaments, made up of a protein that we'll talk a lot about in this class, which is actin. They help generate movement, like in muscle contraction, cell division, cell locomotion, provide for much of the mechanical support, responsible for basic strength and shape of cells, and provide for mechanical support for these cellular extensions. All right, let's call it a day, and we'll pick up and talk about these cytoplasmic extensions that I was already referencing, and we'll get that on Monday.